Hello, this is a quick video talking about macromolecules and the building blocks. So monomers, one piece of a macromolecule, is the building block of that macromolecule. Most of them are built of repeating patterns of smaller bits. So if you think of them like Lego or similar building blocks that you can join together to make a more complex thing, that's the right idea. So a monomer is one small piece, and if it's joined together using dehydration synthesis, you can make a polymer, a long chain or large molecule that is made of repeating patterns of smaller bits. So this is one monomer, that's one monomer, that is. All of these little bits are one monomer, but together we would call it a polymer. And these polymers are, at least for right now, we're focusing on those macromolecules that are important for building cells. These are the types of macromolecules. Here they're called biomolecules, which is another good name. Macro means big and bio means life. They're both big and important for life. So the four types of macromolecule are nucleic acids like DNA and RNA. They're made of units called nucleotides. And you can see a nucleotide has a few different parts to it. And that's okay, because if you have a few different parts that are repeating, here we have green, pink, blue. I don't know what color that is. Green, pink, blue, green, pink, blue, green, pink, blue. We have a repeating pattern. That's the monomer. That's another one. That's another one. So in a case like nucleic acids, that would be the building block is one nucleotide. For our different types of macromolecule, we've got carbohydrates, and carbohydrates, like starches, are made of big groups of monosaccharides. So I'm just going to make a little table down here. For a carbohydrate, the monomer versus the polymer, so the big piece is the polymer, and the monomer is called a monosaccharide. And there's one monosaccharide in particular that we want to memorize, which is glucose. So glucose is the most famous, the most useful for biology. We also have things like fructose and galactose, but I won't be asking you very many questions about those, so you can safely ignore them for now. And the polymer of a carbohydrate, a thing like a starch, or a thing like glycogen, which is what we use to store glucose. Maybe these are our examples. The polymer is called a polysaccharide. So a monosaccharide has one sugar ring, one sweet thing, and a polysaccharide has more than one. We're going to count like this. Mono is 1, di is 2, we'll just go skip right to poly. Poly is greater than 2. That's all. We're going to count 1 too many. That's as complex as we're going to get in our counting right now. So a polysaccharide has more than 2 rings of sugar, like glucose or fructose. For our nucleic acids, the monomer is a nucleotide. The polymer, we're just going to call them by DNA and RNA right now. And the examples are DNA and RNA. We skip over a lot to do with nucleic acids in Bio 20 because in Bio 30 you spend a lot of time talking about them. We've got 2 other types of macromolecule. We've got lipids and proteins left. For lipids, the monomer is a weird monomer. We're going to call it a triglyceride, and this is not a rule set in stone, but a triglyceride has one glycerol molecule as a head and 3 fatty acids. It's joined together kind of like this. Here's one big head with 1, 2, 3 tails. Those tails can either be saturated, which means full of hydrogen, or not saturated, which means there's some double bonds in there. If they're not saturated, they get little bends in them. And they don't stack together nice, and they flow, and they become oils. So the polymer here will be some type of fat or oil, which again will be our examples. And last, we've got our proteins. And the monomer of a protein is something called an amino acid. I'm going to bring these bars down here. The polymer of a protein is called either, we can call it a protein, or we can call it a polypeptide. Peptide is talking about the type of bond between two amino acids. So if you have one amino acid joined together to another amino acid, the bond is called a peptide bond. And that's a special name that we're going to come back to a few times in this course, so it's worth remembering. A peptide bond has, well, lots of unique characteristics, but one in particular is nitrogen is in there. It gives us the right angle for that bond, the right geometry. So examples of proteins are things like ones we will focus on in this course, like fibrin, like actin or myosin, or right now in this unit, all enzymes are things that speed up reactions, I'll make a video about that next, are made of proteins. And some of those enzymes digest lipids or nucleic acids or carbohydrates, and some of those enzymes also digest proteins. So that's interesting. You can make a protein whose job it is to digest proteins. So in this chart here on the side, this just shows the polymer, like for example starch, made of a bunch of monosaccharides, a bunch of single things like glucose. Here we have for nucleic acids, something like a chromosome made of a bunch of nucleic acids. A DNA strand is made of a whole bunch of little nucleotides. And these things, like what's the 5-carbon sugar, what's the nitrogenous base, all that, in Bio 30, we'll spend more time on that. For proteins, things like muscle fibers are made up of strings of polypeptides. Polypeptides are 1, 2, 3, 4, 5, up to maybe hundreds of thousands in a row. These get really, really long, really complex. Amino acids, all of these individual pieces, that's an amino acid, that is too. And for lipids, things like cell membrane, the phospholipid bilayer, or things like just fat cells are made up of things called triglycerides. It has 1 glycerol molecule and 3 fatty acid tails. So those are monomers versus polymers, and hopefully that helps you with your studies.